A very good afternoon to everyone. I hope you all are doing well. Today, in this video, we'll learn about the reproduction, that is for the class 10, chapter number 15. Chapter number 15, reproduction. Unlike the essential life processes such as nutrition, respiration, or excretion, reproduction is not necessary to maintain the life of an individual. Akoi life ama maintain tawbadadi kadawi reproduction je darkar oideba. Akoi life process ta kaidogre kaitamgre. In the chapter life processes, we have learned that for the maintenance of life, we require nutrition, respiration, and the excretion. But reproduction aba process di kadawi akoina life ama maintain tawbadadi darkar oibra process nateba. When an organism reproduces, it creates more individual and a lot of its energy is spent in the process. Then why do organisms reproduce? The organism reproduce to maintain the continuity of their own race. So when an organism reproduce, it help in the organism that reproduce the worker in the kadawi individual masingje hangat haleba. It create more individual, and during this process akon organism ama not organism ni masingje hangat hanba matam da kadawi, it require a lot of energy. So then why do organism reproduce? Akoina kadawi life ama maintain tawbada reproduction je darkar oideba. Adu di akoina organism je reproduce tawibagi maram kaigino haba. The organism reproduce to maintain the continuity of their own race. Akoina kaigumba reproduce tawdadi kadawi akoigi group of organism je extinct oirakaniba. Aduna kadawi akoina extinct oidanaba continue on a sum lainaba gidamakta kadawi organism na reproduce taweba. Reproductions in the plant. The origin of the life is estimated to have developed over 3,000 million years ago and has produced all living organisms of today by different methods of reproductions. Akona kadu life se arte exist waji 3,000 million years ago mamangdagi life se exist in life. So what happened is that before 3,000 million years ago, all the living organism, either it may be plant, either it may be animal, either it may be microorganism, all these organisms are reproduced in or all these organisms exist on the earth by the different method of the reproduction. One of the most important aspects of the living organism is that their ability to produce copy of themselves. According to the organism, the specific character may have. That specific character is that we have the ability to produce copy of themselves. That means according to the human being, the next generation they will have the same features. Their features that means they will have 2 legs, 2 hands, 10 fingers, 1 nose, like that. The next generation will have the same features. 2 distinct methods of producing offspring are found among living organisms. So there are 2 types of the reproduction. One is the asexual reproduction and the second one is the sexual reproduction. So let's see what is asexual reproduction and what is sexual reproduction. Asexual reproduction is a very fast over there. Asexual reproduction is a rapid method of increasing the number of the species without the involvement of the union of the 2 cells, that is the gamete. When a gamete is a haploid male or female germ cell which are able to unite to form a zygote. Adu kadawi gamete habase kaina cell makalabaneba. Kam kanda khona seasonai sexual reproduction ni martamda seasonaiba kadong ba damata new individual produce tawna ba damata adu kadawi asexual method of reproduction di kadawi gamete ani union tawbagi darkar oideba. So in the asexual reproduction, only 1 parent cell is required and it is a very fast method. Sexual reproduction is a slow method in which the union of 2 cells take place for the production of the new individual. In case of the sexual reproduction, 2 individual darkar or 2 cell darkar. to produce the new individual. In asexual reproduction, only one cell is required, and in sexual reproduction, 2 cells is required. Asexual reproduction is a rapid method, and sexual reproduction is a slow method. Now let's learn about the asexual reproduction. In asexual reproduction, the nucleus divides by the mitosis, producing 2 daughter cells. What is meant by mitosis? Mitosis means equational division. Mitosis equational division. In the mitosis, what happens is that the content of the nucleus amasung cytoplasm equally divided into 2 individuals. In asexual reproduction, the nucleus divides by mitosis. Mitosis means division of the content of the nucleus and the cytoplasm into 2 equal parts, producing 2 daughter cells. As the result of the division of the nucleus and the cytoplasm, it results in the production of the 2 daughter cells. In asexual reproduction, the nucleus divides by mitosis, producing 2 daughter cells, each having the same number of chromosomes in their nuclei as that of the parent. Parent number of chromosomes duga new individual the number of chromosomes duga chapmani. For example, parent if the parent have 42 number of chromosomes, then the new individual parents they form the new individual to the they will have 46 number of the chromosome. Cloning. Cloning is a term used to describe the formation of group of the organism of the same species by asexual method involving mitosis and the vegetative or artificial propagation. Cloning is also method used for the formation of the group of the organism of the same species. In the cloning, the new individual may be produced by asexual reproduction, which may involve mitosis. Mitosis means divisions of the nucleus and the cytoplasm into 2 equal parts, or vegetative or artificial propagation, or we can use different part of the plant for the production of the new individual. The clone have identically genetic composition. That means the clone na di kaina akoina phangiba new individual the yauriba genetic composition sing do kaidoi parents the yauriba genetic composition duga chap manana akoina phangniba. This method is widely used by microorganisms, plant, and the lower animals. So the cloning process is used for the productions of new individuals, and it can be performed in the microorganisms also, in the plant also, and in the lower animal also. The next topic is the budding. Yeast cells form a small bud into which the cytoplasm and the nucleus passes. The bud is much smaller than the parent cell and become detached. Yeast is a unicellular organism. That is, the yeast is made up of only one cell. So let's see. In the yeast, this is the parent cell which contains the nucleus. So when the parent cell get mature, what happens is that new outgrowth develop. Parent cell mature and the small outgrowth develop, and that small outgrowth is called the bud. The content of the cytoplasm and the nucleus of the parent cell started splitting into the new bud. So once the splitting of the nucleus and the cytoplasm is completed, what will happen? The bud, new outgrowth develop the new outgrowth or the bud will detach. Parent to the parent cell to the the bud will get detached from the parent cell and the new individual will develop. This is how the yeast produce the new organism. A multicellular organism like Hydra also reproduce by the formation of the bud, which later separate from the parent cell. In unicellular organism like yeast and in multicellular organism like Hydra, both the organisms reproduce the new individual by the method of the budding. The next is the fragmentation. In the multicellular filamentous algae like Spirogyra, a parent cell divides by forming new cell wall or the septum into the daughter cell. That is septation or the fragmentation. So, in the Spirogyra, what happens is that when the parent cell mature, there will be formations of the septum, also known as the cell wall. So, parent cell mature septum developed or ever. So, once the septum is complete, or once the development of the cell wall is complete, this will form the new individual. This is how the new individual is developed in the Spirogyra by the method of the fragmentation. Sporulation, spore formation. Fungi, liverwood, mosses, and the fern can produce enormous number of the spore. Spore are the small bodies containing a nucleus and a small amount of the cytoplasm. So, spore can be defined as the small bodies. Bodies containing a nucleus and a small amount of the cytoplasm. The spore of the terrestrial organism are usually very light. Spores are very light and have a protective wall around them. And this wall, spore wall, it helps in the protections against the unfavorable environmental condition. Many bacteria form a thick wall spore inside the bacterial cell. So inside the bacterial cell, there is presence of the thick wall spore. This cannot be considered as a reproductive mechanism for them, but provide a means of the survival during unfavorable conditions. Sinahabadi ka no, ngasagi yauriba thick wall spore do na kadawi reproduction da di mateng pang deba, but ngasagi thick wall spore sina kay da mateng pang habada, it help in the survival of the spore during the unfavorable condition. Some of the green algae and the aquatic fungi form 2 or 8 small zoospore. Zoospore, spore of the algae, fungi, and the protozoa. Some of the green algae and the aquatic fungi form 2 or 8 small zoospore, each with a nucleus. So the zoospore contain, or the spore of the algae, fungi, and the protozoa contain nucleus, cytoplasm, and the 2 flagella. Swimming by means of their flagella, the spore disperse the species to the new location. So, the flagella, which is present in the spore of the algae, fungi, and the protozoa, help in the swimming in the water bodies. And during the swimming, or with the help of the flagella, what will happen? The spore will be dispersed to the new location. They will settle in the new location and they will reproduce in the new location. Activity number 15.2. Keep a slice of the moistened bread in a moist, warm, and the dark place. Observe the slice daily for a week with a magnifying glass. Record your daily observation. So what we can do is we can take a piece of the bread. Ako na bread ki piece ama lower din. Then we have to keep the piece of the bread outside where there is moisture. Ising yaw ba chocho law ba mapangumbam da kadoy ako na bread cha thamani ba. Then we have to observe the slice of the bread every day. Then after some days you will see that a layer of the white or the gray structure developing on the bread. Ako na bread chika doy hapta ama matung da kadoy ako na white structure ama uiba natraga gray structure. That gray structure is the rhizopus. So let's study the thread-like structure that develop on the bread in the activity 12.2 are above are the hyphae of either the bread mold rhizopus or the pin mold mucor from its body or the mycelium. So the structure which is developed in the bread may be either rhizopus or it may be either mucor, and the body and that structure developed from the body which is known as the mycelium. The erect hyphae develop a swelling at the tips to form the sporangium. So this is the structure of the rhizopus, and at the tip there is presence of the sporangium. Inside the sporangium, a large number of the spores are produced by the mitosis and the liberated. So, at the tip of this structure, there is presence of the spore inside the sporangium. So, this spore is produced by the process of the mitosis, and this spore, when this sporangium mature, what will happen? The spore will be released, the spore will be settled down, and what will happen? From this spore, new organisms will be developed. This is how different organisms develop from the spore. The mosses, clump mosses, and some common ferns are also produce enormous number of the tiny windblow spores, which serve to display the species to the new location. So what happened in the mosses is that the spore will be flow, or the spore will fly away with the help of the wind, and they will land in the another new location on the land torniba, and in that location the new organism will start developing. The next is the vegetative reproduction. Vegetative reproduction is a method of the asexual reproduction. That means there will not be involvement of any two gametes, only the parent cell will be involved. Vegetative reproduction is a method of the asexual reproduction in which the vegetative part, such as root, stem, leaf, and the bud, can be detached from the parent. Detached adikadu akoina thektok pa haibado. Natraga akoina kadawi root, natraga stem, natraga leaves, natraga bud se kadawi akoina parent cell si they lawrani ba. Aduga kadawi from that the new organism will be developed. Can be detached from the parent plant body and develop into the another complete daughter plant. So in the vegetative reproduction, what do we use is we use the different part of the plant. Plant took a different part. Either it may be stem, either it may be root, either it may be bud, or either it may be leaf. From that, the new organism will be developed, or the daughter plant will be developed. This method of asexual reproduction is seen in many flowering plants like sweet potato, onion, potato, grasses, banana, etc. So vegetative means of the reproductions can be performed in the flowering plant such as sweet potato, onion, potato, grasses, banana, etc. Perennial plant. Perennial is the plant which grow for more than 2 years. 2 years they have hingbang plants in the book perennial plant and the perennial plant which over winter di kadawi winter gi matam dan hena kadawi moje growth lebado natraka spread obado. Perennial plant which over winter as bulb, combs, tubers, or the rhizome are the vegetative propagated. This property of vegetative propagation is used artificially by the method of layering. Layering means development of new plant from the young branch. Grafting means the stem is cut, the cut stem is injected into the stem of another plant so that the new plant will have the quality of both the plant. Cutting means we can cut any part of the plant, and that cut plant will develop into the another new plant. And by artificial culture in the different media, or we can develop new plant by culturing in the different media also. Artificial propagation is successfully performed in the plant like rose, orange, grapes, sugarcane, banana, jasmine, etc. Another disadvantage of the vegetative reproduction is that the offspring produced are genetically identical to the parent cell. So the new organism which is developed by the means of the vegetative reproductions are genetically identical to the parent plant. The next topic is the micropropagation or tissue culture. It is the technique of propagations of plant. Propagation hai dekho multiplication ga hai. It is a technique of propagations of plant from the cell, tissue, organ by using the method of the tissue culture. So, by the method of the tissue culture, we are going to multiply, or we are going to develop the plant from the cell, from the tissue, or from the organs. Here, the Plant parts, root, stem, leaf are cultured on a sterilized medium containing the necessary nutrient. So first we'll take a medium which contains the nutrient, and in that medium we'll culture our cell or the tissue or organ or any part of the plant. After some days, a mass of the undifferentiated cell known as the callus is developed. So when the cell or tissue or organ is cultured in the sterilized medium which contains the nutrients, there will be development of the callus. By transferring the cell of the callus to the another medium containing the hormones, the callus differentiate into the plantlet. So once the callus is developed, what we have to do is we have to transfer the callus again to another media which contain the hormone, and these hormones help in the development of the plantlet or differentiations of the plantlet from the callus. Here first we'll take a media which contains the nutrient, and the cell or the organ or any part of the plant will be cultured here. Then after some day we'll observe the development of callus, and this callus is again transferred to the media which contain the hormones, and after that we'll see the development of the plantlet. Once the plantlet is developed, it is transferred to the soil and new organism is developed. This is how the tissue culture is performed. This method is successfully used in many economically important plants such as orchid, chrysanthemum, gladiolus, etc. The ability of the somatic cell to produce the complete organism is called the cellular totipotency. It was first observed in the carrot cell. The next is the parthenogenesis. Parthenogenesis is the type of the asexual reproduction in which the organism is developed from the unfertilized egg. So, development of the new organism from the unfertilized egg is called the parthenogenesis. This process may be natural or artificial. It can be done in the laboratory, or it can be a natural process. Natural parthenogenesis is found in many plants and the animal. Non-flowering plants like Spirogyra, Cara, Marcelle, etc. and the flowering plant like Thalictrum and certain species of the Asteraceae, sunflower families, and the Solanaceae, tomato family can reproduce by the parthenogenesis due to the absence of the flowering plant. The flowering plant is the reproductive part, but due to the absence of the flowering plant, in some organisms, the new individual developed from the unfertilized egg. Some examples are given here. In animal, this process is found in the aphid or the green fly, but they are normally reproduced by the sexual reproduction.